Good afternoon, everyone. My name is Troy Jones, and I am a senior biology major at Morehouse College. And today I will be presenting my research concerning fenugreek and its ability to show selective cytotoxicity against the multiple myeloma cell line. Beginning with establishing some context, multiple myeloma exists as a cancer of the bone marrow, which possesses a lot of implications as far as the human immune system is concerned. So what happens is your bone marrow is responsible for producing these plasma cells, and these plasma cells go on to produce antibodies. Now these antibodies serve as your first line of defense should a foreign antigen enter the body. However, in the case of multiple myeloma, what happens is you have these compromised plasma cells, which are then converted into abnormal plasma cells. And what this does is instead of producing functioning antibodies, it produces defunct antibodies known as M proteins. Now these M proteins don't cause harm necessarily to the body. However, because they're defunct or inactive antibodies, it ends up taking up space for the normally functioning antibodies to do their job, which would make it that much more difficult for the human body to produce an immune response should an antigen present itself. Now, regarding multiple myeloma and its impact socially, it's the second most common hematologic malignancy in the United States. Individuals aged 65 and older are usually the ones more afflicted by this. And also noteworthy is that African Americans are 2 times as likely to be diagnosed with multiple myeloma compared to their white counterparts. The 5-year survival rate doesn't look very well. There are 0 curative treatments for this disease. So within being diagnosed, and then 5 years in between that, all the individuals diagnosed on that initial, probably 37% of them are still living after 5 years. So to kind of just bring this all together, I'm going to introduce our hero, which is the fenugreek, right? So fenugreek has been reported to contain a host of human health benefits, but what we want to focus on is its potential to be an anti-cancer agent, right? So what we hypothesize is that the fenugreek contains chemicals that have anti-carcinogenic properties, which would therefore cause cancer cell death, and we therefore predict that the viability of multiple myeloma cells will decrease when exposed to whole plant fenugreek extract. Now, the reason why we chose to move with fenugreek is because previous literature has already quantified fenugreek's ability to induce apoptosis in MCF7 breast cancer cells. So in hopes to further contextualize fenugreek as an anti-cancer agent, we wanted to see if it displayed a similar effect with various cancers. So in order to do so, the first thing we had to do was produce a viable extract that we know produced an effect on the cells. So to do that, what we did was we took the fenugreek seed and planted it under greenhouse conditions in neutral soil for a period of 20 to 30 days to allow for maturation. After this maturation period, the fenugreek plants were harvested and dried. The whole plant was harvested and dried at 50 degrees Celsius for 24 hours before it was sent over to a Wiley mill for grinding. And the purpose that we use for grinding is to convert the plant material into a homologous structure, so that when we move on to our next step, which is the methanol extraction process, where we mix the plant material, depending on how much plant material is yielded too, so it varies, but with a given amount of methanol, and the two contents are made to mix for a period of 2 hours on a hot plate at a temperature, so that way we can evaporate that methanol off. And so moving forward to our experimental design, MM1S cells, or multiple myeloma cells, were held in culture before being subject to varied concentrations of the fenugreek extract for a period of 24 hours. Upon this 24-hour period, upon completion of this, the cells were then harvested and prepared for a 2-color live dead viability assay, which is an instrument or a technique that we use in order to distinguish living cells within a population from the dead ones. So as a result, it was determined that the fenugreek extract in turn produced killing. This was replicated over 3 times, and in saying it was replicated over 3 times, I mean the entirety of the process. So that means extract production was replicated 3 times, the culturing experiments where they were subject to the extract, that occurred over 3 times, and then we've seen the results, which told us the extract was in fact doing its killing. So the next question that arose in conducting this study was whether or not this killing was selectively cytotoxic. Okay, we didn't know whether or not this extract just killed cancer cells specifically, or if it had the potential to also affect normally functioning cells. So in order to determine this, we introduced two new cell lines, and that would be C2C12 and HEK293 cells. And what we found in applying the same experimental design with these normally functioning human cells was that in the face of the extract, when exposed to the extract for the same amount of time, there was relatively no change in the viability, which was able to solidify to us that the extract produced was in fact demonstrating a selectively cytotoxic effect on the myeloma cell specifically, or on the cancer cell specifically. So in conclusion, the fenugreek extract proved to be effective in killing the cancer cells, killing over 50% of the myeloma cell population at the greatest concentration listed. And also the extract apparently shows selectivity in its killing because it was non-lethal at the examined concentration for the non-cancerous cells. So broader impacts and also future directions for this study. Perhaps the most immediate plan of action is to determine the mechanism of action by which this fenugreek extract is doing its killing. We don't know if it's inducing apoptosis, so that would be sort of the next, I think, step in also contextualizing fenugreek as an anti-cancer agent. As mentioned previously in previous studies where fenugreek seed extracts were applied to breast cancer cells, the investigators of those studies determined that it did in fact induce apoptosis, which is informing my prediction that in the case of the multiple myeloma cells, we'll probably see a similar effect. Further down the line, we would like to see what component of the fenugreek plant too, depending on whether it's the root or the leaf or the stem, considering that our extract for the purpose of this study was the whole plant. So we would like to sort of hone in on what component of the plant is responsible for inducing that apoptosis. I would like to acknowledge the Morehouse College McNair program, trio, and my mentor, Dr. Handy. Yeah, thank you for tuning in.